this is work done by Carlos Pardo, who's a neuropathologist and neurologist at Johns Hopkins. And he was enticed into autism research by Andy Zimmerman, who's a pediatric neurologist there, who's been interested in the immune system and autism for quite a while, and felt that we really had to look at brain tissue. So Pardo had experience looking at conditions in adults with known immune problems. I think he was doing Alzheimer's, Huntington's, and or AIDS, I forget which exactly one. But the point was he knew staining techniques which would allow you to see immune activity. And he found these little dots in there are activated glial cells. Glial cells have an immune function, so there's the astroglial cells and the microglia. Astroglia, well, microglias are related to blood cells, but they get turned activated when they're having to do immune work. And they shouldn't be that activated under normal circumstances. So to find this in brain tissue was different than what anybody had found before. And it changed, in my view, the ball field. Whereas before, it was 2D, genes, whatever. Now you had to say, it's the tissue has something going on. And what he found was these brains, it's very hard to get the brain tissue. And that's why there's campaigns that if you have some unfortunate accident with your loved one with autism, you should donate the brain to a tissue center, because it's very hard to do research like this if there's no samples, and it's very disturbing when someone passes away. And it's hard to think about, but that's the only way we find this stuff out. So they were able to get 11 brains, which is not a lot, from 5-year-old to 44, and they got not the whole brain, but just 3 regions. An area here, the middle frontal, an area in the middle called the anterior cingulate, and part of the cerebellum. 5 years old through 44, and they were death by various reasons, including drowning, which is a bit of a problem, which I'll explain. And all of them had this activated glial cells. And this finding has been very disturbing to some of my colleagues. Well, some of them dismiss it and say, well, you can get this after birth, after death, if you don't treat the tissue right, which isn't true, because you need oxygen. If they don't have it in the first place, they can't develop it after they die, although the ones who drowned, maybe they weren't quite dead immediately, and it's possible that they could have developed them. What was interesting to me, Pardo invited me down to his lab, and I gave a talk down there, and he took me in, and I was looking at this stuff under a microscope. But one of the things I wanted to know was, from 5 to 44, does it get worse or does it get better? And the answer is neither. It basically was about the same intensity from 5 to 44 years old, but he got a 3-year-old brain from Eric Crochane, and that brain had a lot more activated microglia. And that, then you're going, well, I already have it in my head that these brains get really big really fast. And this kid who was younger had a lot more. Is this what's making the brain big? Well, it certainly is the primary question in my mind, but I just have to say to you it hasn't been proved yet, and what we need now is experiments to decide whether it is or not, and I'm going to show you some of the things that have come along so far. But first I want to point out, this is an article by Lillian Calderon. I had her come speak at the Autism Society Science Symposium last summer. She is not an autism researcher. She was studying air pollution and the brain in Mexico City, where there's awful air pollution, as you probably know. And she got 2 studies. One of them was on children, comparing children in Mexico City with children in rural Minnesota, where she teaches, and who died abruptly, getting hit by a car, scooter accident, something very sudden, no history of neurological, neurodegenerative. They had changes in the brain with inflammation, blood-brain barrier disruption, ultrafine particulate matter deposits, and ultrafine particulate matter is part of what's in air pollution. You get it from catalytic converters, which were supposed to be helping air pollution, but they only created a different kind of air pollution. It's harder to see. An accumulation of alpha amyloid beta 42 and alpha-synuclein, which are related, which are what you see in old people with Alzheimer's and Parkinson's disease. And here you're seeing it in children and very young adults. And she also did a dog study looking at dog brains and found the same thing. And she found chemokine and cytokine immune chemicals, which were very. So what happened was I invited her to this meeting, and I sent her Pardo stuff and a bunch of other stuff. And by the time she'd gotten to the meeting and prepared her talk, she was talking about autism because she'd done some homework, and she said pretty much what she's finding both at the tissue level and at the immune chemical level is extremely similar to what's found in autism. And it's really important to realize that what we're seeing here is not that specific to us. There's another guy, there's somebody else who's a very interesting researcher at the National Institute of Aging, Kevin Becker. He sent me some work a few years ago about brain enlargement in children with allergies who don't have autism. He has a new paper that's just come out on Tylenol as a problem in autism and asthma. Very interesting thinker. He came to my talk with Hopkins. So here's my picture again with the outer white matter being enlarged and the inner, the white part, being not enlarged. So what Pardo showed me the day when I went to visit him was that he had, my paper came out while his was in press, and so he didn't have a time to do this investigation for that paper, but he went back to his brains and looked at the outer compared to the inner white matter to see if there's a difference in inflammation, and he found that the outer area right here had astroglial activation and the deep white matter did not. And there was a lot of microglial activation out here in the gray matter. You can see it looks great. This is actually a picture of brain tissue. It's not a scan. And that was the idea that you could see in a scan, which doesn't show you cells, something which is related to something you can see under the microscope. And that was my work, and that inspired that. That was very moving to me. But it does raise it kind of supports that it is important to be going to look to see whether this white matter here is inflamed or not. Now, a lot of people were assuming that this white matter was more of those wires, so that the kids had better connections in the short areas one place to another, but worse in the long-range connections. And they were making up all kinds of theories that explained how good people with autism can be with small details and how bad they can be with hooking things together. But that theory depends on it really, that white matter enlargement actually having wires in it. Or that, of course it has wires in it, but having more wires in it. This is a paper that came out now 3 or 4 years ago, and it showed that there are areas of the brain, the yellow parts, which are very similar to what the areas, many of them, to where we found enlargement, and where Pardo found that inflammation. And the change that was recorded in this study was consistent not with more myelinated axons, with more fibers, but with more water. So it's almost like either swelling or the cells can't pump water properly, which could be a mitochondrial problem. And there's subsequently been, I don't know how many more, so I don't have more, but there have been a number of other studies looking with other techniques to see what the properties are of the tissue there. And they're finding by other techniques that it doesn't look like it's wires, it doesn't look like it's more cells, it looks like it's less cells, more water, and more space. So it's more consistent with an inflammation or an oxidative stress or a mitochondrial problem than it is with a cellular overgrowth problem. So this is why I do brain imaging research, is because there are still people who don't want to think about this and just are assuming that it has to be another way, and the way you test that is you test it. And we have to know at the level of science, and also this really informs public policy and what we do about this.